In biology, when we use the term cloning, we mean the production of genetically identical copies of DNA, cells, or organisms through asexual means. So when an underground stem or root sends up new shoots, the resulting plants are going to be clones of one another. When you grow colonies of bacteria in a petri dish, you are typically looking at groups of clones because the whole colony grows out of a single-celled organism. Identical twins are an example of human cloning, since a single embryo divides in two. The first time cloning was ever performed in a scientific venue, it was done on living organisms. The process was initially called twinning because that's what it is. It is the forced, I guess, the forcing of genetic twin happening. So in other words, I just want to point out that the term clone can mean a lot of things, and it's actually a very common process used in technology all the time. So biotechnology is the field in which scientists make use of controlled living cells to perform specific tasks. We're going to do this through genetic engineering. A subset of biotechnology involves the manipulation of DNA for human purposes. Some uses, there's a lot of reasons why scientists may want to clone a particular gene. And one of the things we're going to talk about the most is going to be the cloning of genes and or the synthesis of proteins. But some of those uses might be to produce drugs and hormones, improve treatments for human diseases, increase food production from plants and animals. When clone genes are used to modify other individuals, the result is what we call a transgenic organism. Transgenic organisms can be very valuable to humans. For example, taking a gene from a desert-thriving form of rice in the Americas and splicing it into a species of wild rice native to Africa can produce a drought-resistant strain that could triple rice production and greatly help impoverished areas around the world. So this process that we are going to use to come up with these transgenic organisms is using a technology known as recombinant DNA. Recombinant DNA contains DNA from two or more different sources, like human and bacterial DNA. The benefit of this can be huge. So a great example of this would be the production of insulin. So I'm not sure how familiar you are with the production of insulin, but this is actually a really cool process. Human insulin is a complex protein that is very difficult and very expensive to make in the lab. But do you know what bacteria is really good at? Bacteria is really good at making proteins. Bacteria, prokaryotic cells, are full of ribosomes floating around looking for proteins to synthesize. So what we have managed to do, and we're going to talk about this process, is insert the gene for making insulin into bacteria, and then we can turn bacteria into little protein factories that are going to make mass quantities of insulin for us that can be easily harvested and put to good use. So there are going to be several things that are going to be required for this particular feat of engineering. First, we're going to need a vector. And you know what? Actually, I have a lot of slides about this, so we're going to just go right into the next slide. And I'm going to, we're going to start off with this little video that looks at recombinant DNA and how it works.
The basic idea behind genetic engineering is to join DNA from two or more sources. In this case, one piece of DNA is a gene of interest from a eukaryotic cell. The other is a circular DNA molecule called a plasmid that originates from a bacterium. A region of DNA from any source typically has numerous small nucleotide sequences that can be cut in a test tube by a variety of enzymes called restriction enzymes. The restriction enzyme EcoR1 cuts DNA at the recognition sequence GAATTC. Note that the enzyme produces a staggered cut, leaving free single-stranded ends called sticky ends. The sticky ends have a tendency to pair with complementary sticky ends on other molecules. The ends are pasted together permanently by another enzyme, DNA ligase, resulting in a piece of recombinant DNA. The recombinant DNA is transferred to a new host cell. The plasmids, which originate in bacteria, allow the recombinant DNA to be propagated in bacterial cells as the cells reproduce. Each colony of bacteria is a clone, a group of genetically identical organisms, all descended from a single cell. Bacteria containing the plasmid with the gene of interest can be identified and grown in huge numbers, and the gene can then be harvested and analyzed. The basic idea.
There we go. So now we're going to go through those processes individually, starting off with the steps in creating recombinant DNA. We're going to start off by using a restriction enzyme to cut the gene of interest out of the original DNA and splice it into the vector DNA. So in this case, we're going to cut out our gene of interest. Let's just say this is our sequence for making human insulin. So we're going to cut out that section of DNA. We're going to then open up our vector DNA, insert our DNA of interest into it, and then move on. So the plasmids are going to be common vectors. Plasmids are circular pieces of DNA that exist separately from bacterial chromosomes. If you remember way on back when we talked about prokaryotic cells, we talked about where they keep their chromosomes. Does anybody remember where bacteria keep their DNA? That's right, they keep it in a nucleoid region. It's a region of the cell where that DNA just sort of hangs out. Now that's going to be their chromosome. That contains all the information that bacteria need to be a bacteria. However, bacteria have these things that are known as plasmids. Plasmids are circular pieces of DNA that are extrachromosomal. They're not necessary to the overall function of the bacteria, but they can be helpful. For example, if some bacteria discover that they have a resistance to a particular antibiotic, they can share that DNA, that information back and forth with other bacteria through plasmids. So bacteria are actually uniquely designed to pick up DNA from their environment and start replicating it. A vector is going to be, that recombined plasmid is going to be taken up by that bacteria, which will be our new host cell. The bacteria with the gene of interest will then have to be identified and isolated, and then it's going to produce numerous copies of that gene or amplify that gene through a process known as bacterial cloning. So not only is the bacteria itself going to reproduce, and each time he reproduces, his offspring is going to have that particular bit of plasmid, but also it's going to start synthesizing that plasmid, start synthesizing that RNA into DNA. So we can produce numerous copies of that gene through either bacterial cloning, or we can artificially create numerous copies of that gene ourselves through what is known as a polymerase chain reaction, PCR. Here is a diagram that shows us how we can create that or multiply that recombinant DNA using bacterial cloning. You should be able to, by the end of this chapter, put your hand over the text on the left-hand side and walk through this entire process, knowing what's going on. We're starting off with our plasmid and our source DNA. We then need to cut out our specific source DNA, insert it into our bacterial plasmids, insert our plasmids into our hosts, or insert our recombinant DNA into our hosts, and then allow that host to clone the copies of that gene, and then also start producing or synthesizing those gene products. Here is an example of the PCR cycle. PCR, polymerase chain reaction. This is a way that we can artificially produce or we can artificially clone mass amounts of DNA very, very quickly. This is not going to require the aid of a life form like a bacteria. This is actually just a little countertop machine about the size of a small toaster oven.
Let's examine the basic PCR cycle. Temperature changes are automated by PCR machines called thermocyclers. The reaction mixture contains the target DNA, DNA primers, DNA nucleotides, and a heat-stable DNA polymerase called Taq polymerase. In the first step, denaturation, the solution is heated to nearly boiling, 95 degrees Celsius. The heat breaks the hydrogen bonds between the two DNA strands and allows them to separate. In the second step, primer annealing, the temperature is dropped to around 60 degrees Celsius. The exact temperature depends on the length and base composition of the primers. The primers are synthesized so that their sequences match the flanks of the target DNA sequence. At this relatively low temperature, the primers can form hydrogen bonds with the single-stranded DNA, a process called hybridization or annealing. The primers are present in great excess relative to the target DNA, ensuring that most target strands anneal to a primer and not to each other. In the third and final step, extension, the temperature is increased to 72 degrees Celsius. A heat-stable DNA polymerase called Taq polymerase, which originates from the hot springs bacterium Thermus aquaticus, functions optimally at this temperature and is not destroyed at the denaturation step. The polymerase now adds additional DNA nucleotides. Each new DNA nucleotide that joins the growing strand is complementary to the nucleotide in the opposite strand. After an appropriate incubation period, there are 2 DNA copies instead of the 1 original copy. In cycle 2, the process repeats, so that there are now 4 double-stranded copies. Each PCR cycle literally doubles the content of the original target DNA. In practice, 20 to 30 cycles are usually run, yielding a 10 to the 6th to 10 to the 9th fold increase in the target sequence. Roll your cursor over the bars in the histogram to see how many DNA molecules are theoretically produced by the end of each PCR cycle. Assume that 1 double-stranded molecule exists at the beginning of cycle 1.
So this is just some text to go along with that. This is how the polymerase chain reaction works. It's going to amplify a targeted sequence of DNA, and it can create millions of copies of genes or a specific piece of DNA in a test tube. It requires very little. It requires DNA polymerase that can withstand the temperature necessary to separate double-stranded DNA, a supply of nucleotides for a new complementary strand, and the amount of DNA is going to double with each replication cycle. The last thing we're going to talk about with genetic engineering is some of the applications. There are so many applications for genetic engineering. There's a reason why we do this. Some of those applications include environmental. So, for example, by genetically modifying oil-eating microbes that could withstand ocean conditions. They naturally consume oil. If we could get them to withstand the salty conditions of the ocean, we could have microbes that could be released into the ocean to clean up after oil spills that could then become plankton to feed other microscopic animals themselves. Livestock, for example, cows could be genetically engineered to produce more milk. Pharmaceuticals, gene farming, create transgenic animals that produce a protein with medicinal value in either their milk, eggs, or blood. We just then have to collect and purify the protein. Raw materials. For example, goats and bacteria have been genetically engineered to produce spider silk proteins. So goats that can produce the spider silk, it can be obtained through their milk. You just milk the goat and you simplify out, or you isolate those spider silk proteins from their milk, and that can be then used to make any sort of strong and lightweight fibers. Agriculture. For example, crops can be genetically engineered to be resistant to pests. So here's just an example of how a goat could be genetically modified to produce a protein of interest. Gene therapy is another very important use of gene engineering. Gene therapy is where we can put functional genes into body cells that have been affected by a defective mutant. Viruses are fantastic. Viruses are amazing gene delivery systems. This is how they function. Viruses are little more than a hard protein shell around a code of DNA. They then inject that DNA into our cells and trick our cells into making millions of copies of their own virus that can then spread out through the rest of the body, infecting all of our cells as they go. However, if we could hijack that system, empty the virus of whatever harmful DNA it's containing, and inject the DNA that we need for the missing protein, we could then potentially start synthesizing and making that protein, making that missing gene that we need. So it has the potential to cure inherited diseases. There has been some success with severe combined immunodeficiency for this particular type of gene therapy. All right, so you should be able to, at this point, go through all of these different steps. These should be your goals for this chapter. I'm not going to read through them all because you can do that on your own. All right, that's all I have for this chapter.